Welcome back to the next session in BOSC, which is on data visualization. So it's sure to be a really interesting set of talks. So I'd just like to mention that I joined the organising committee this year for BOSC. So it's been really nice to have such a welcoming group of people and such an awesome kind of group of people yourselves to join and kind of get to know. So without further ado, I'll pass over to Anthony, who will be talking to us about WormGUIDES. So.
Yeah, I think I'm good. Hi, it's good to have an opportunity to talk about WormGUIDES, which is an ongoing project to try and build an interactive, single-cell level atlas of C. elegans development. So this is a multi-institution project that's split between a number of labs. Daniel Colon-Ramos' lab at Yale, focused on neural development. Harry Schroff at the NIH, who's handling the microscopy end of it. Our lab, the Bao lab at Sloan Kettering, we work in developmental biology, computer vision, visualization, and Bill Moeller's lab at University of Connecticut with a focus on developmental biology and visualization. So when I'm talking about an atlas, what I mean in this context isn't just a bunch of images, which is what people tend to think of with an atlas, but images that are hung on a scaffolding of sort of more abstracted representations of the embryo. Nuclear positions, where every cell is at the level of the position of the nucleus at every point in time over the whole of embryonic development, as well as the details, ultimately at least, of neuromorphology over that whole course of development also, as well as additional annotation like the wiring diagram of the adult worm and any other sort of information from the worm community or from individual users. For those who aren't really familiar with C. elegans, it's sort of the perfect and perhaps the only at the moment place to try and do this because the genetics tools are very mature. It's got a very easily observed development. It's relatively small. It's nice and transparent. It's the only organism where the neural anatomy is known in detail at the wiring diagram level. And famously, or if you're not worm biologist, maybe not, is the fact that it has an invariant lineage. So in every single embryo, there are the exact same number of cells, and they all do the exact same thing, which very critically makes the superimposition of multiple data sets on top of each other tractable. So there's a bunch of moving parts in this project, and I want to start by talking about some of the open source projects in the different contributing labs that are sort of becoming part of WormGUIDES before going on to the big vision for WormGUIDES itself and sort of what we've done already. So a key part of the pipeline, and I'm not saying that just because I'm the primary developer on it, is Starry Night, which is a cell lineaging pipeline that takes you from a movie of development like the one you're watching on the top to an abstract representation of where each cell is at each point in time, a geometrical representation like the movie you just saw right below it. Without going into sort of details about algorithm and the like, it's a piece of software that's really optimized for doing robust cell tracking in images that are crowded and undersampled axially, where you don't have as many slices as you'd like, which is really critical for doing this kind of analysis where you want to track in a reasonable amount of time with perfect accuracy hundreds of embryos over hundreds of time frames, hundreds of cells. Starry Night is pretty closely related to ACE-Tree, which was originally developed in the Watterson lab at University of Washington, is currently scattered among a bunch of places, but we're one of the labs that's contributing to it, which is a visualization tool for doing single cell analysis, for looking at the images and the abstract tracking results together, doing editing if necessary, looking at gene expression over time within the nucleus, and we're actually trying to build that into a more sort of community-based platform for plug-in based data analysis beyond the functionality it already has. Another contributing project is SaitoShow, created by Bill Moeller at University of Connecticut. It's a web app meant for sharing image data in large data sets, but more critically, annotation at the single cell level over the web as a sort of web start Java app. I want to switch focus now to WormGUIDES itself, briefly talk about what the final vision we have for what's going to be in there, and then talk about what we've got so far. So the long-term project goal with WormGUIDES is really to take many image data sets with different structures highlighted in them and superimpose them all on top of each other, both as raw image data and as abstractions in the form of nuclear positions and segmented individual neurons. And the general strategy that's going to be followed is doing two-color imaging. On the lower left, you can see an embryo where all the nuclei are labeled in red, and a subset of neurons are labeled in green. On the right, you're looking at a rotation of that same neural structure segmented from a data set like that. And so the idea is that at the scale of hundreds of embryos, using different strains with different promoters that label different subsets of neurons, you'll be able to segment each individual neuron relatively easy because the images aren't very crowded, and then superimpose those different data sets using the nuclear positions as a guide, because you know not just where each nucleus is, but you actually know which individual cell identity each one has, because you're tracing them from the beginning, minute by minute. And this part of the project is really at the preliminary stages. So I'd love to talk to people about detail-oriented stuff before we get it all wrong. There's a lot of open issues in terms of the computer vision for doing the segmentation in a high throughput fashion. At the moment, we're doing preliminary results more or less entirely manually. And also in terms of the alignment. But what's actually more interesting to me as someone whose background is in graphics is sort of what are the right paradigms for trying to explore the data and be able to extract useful information from these really complicated scenes. Even in the abstract form, 300-odd neurons among 600-odd cells, it's a lot of stuff going on, and it is kind of complicated to navigate. Speaking to that, what we've got so far is a concrete thing you could actually download and try and play with is the WormGUIDES mobile app. So it's a mobile application available on iOS or Android that lets you browse the development over time just at the level of detail of the nuclear positions. So it's kind of hard to tell from static images, but you can scroll back and forward in time from the 4-cell stage to about 500-cell stage of the developing embryo, see where any given nucleus is, and sort of in a spatially oriented way, pick a cell that's of interest to you, tap on it, and you'll come up with a window like that, which will tell you what the final fate of the cell is, and will let you then query worm community databases from more or less whatever is known about that particular cell. So a big part of the goal for the application is to make it possible for biologists to generate customized displays or visualizations that highlight phenomena that are of interest to them. If you kind of are or deal with worm biologists, they always seem to have their favorite cell that they sort of want to know all the intricacies of what it might do and who it might be interacting with. And so what you can do here is in a relatively easy search interface, you can highlight individual cells, you can highlight cells based on their final fate, or any keywords in the standard parts list. You can also query Wormbase for the cells that are known to express a particular gene, and you can overlay all that information in different colors on top of the scene to try and create a custom visualization highlighting what might be sort of important players in the phenomena that you're interested in. And, you know, especially with the genetic information overlaid on top of it, hopefully start to be able to generate hypotheses about sort of mechanistically what's happening. Again, it's kind of hard to tell from static screenshots what's going on. I encourage people to try and download the app if it's even slightly interesting. So this is what we have at the moment. The next step in really the next couple months is to actually start to superimpose a little bit of neural morphology data on top of this from a couple of data sets that we were starting to generate using a more or less manual pipeline. And I think we're going to learn a lot when we start to do that about what the issues of some of the visualization issues of trying to like navigate. Even the nuclei are complicated enough once you start to layer in a bunch of colors, but to navigate the much more complicated display when you start to put in detailed morphology and look at how the shapes of multiple nearby cells are interacting with each other. So that's what's coming up. We'd love input on that. I want to mention that even if we actually, not if, but when we accomplish all of that, that's actually just the beginning. This is a movie created with our cell tracking pipeline of the development of a Drosophila embryo, and there's a whole host of even more complicated issues about how you would create an atlas like this when you don't have the scaffold of the invariant lineage to essentially allow you to cheat to semi-trivially superimpose multiple data sets on top of each other. With that, I'd like to say that we really welcome at this stage input or collaborations. We're really interested, I think probably everyone here knows that, like most sort of open source academic projects, it's open source in the sense that you can easily download the source code and compile it should you want to, but I can probably count without taking my shoes off how many people have actually done that. Most users use it at the application level, but we are really interested in trying to figure out, because we certainly don't know how to actually make it a real open source, sort of more community-based project. Thanks.
So, yep, great, thanks, Anthony. Does anybody have any questions? There were very few from the last session, so I want to see some more questions.
I'll make up answers.
Okay, well, I have a question. So data integration is typically hard, right? So you kind of alluded to the fact when you've got a really nice kind of system here for looking at that kind of data integration. Yeah, you've kind of got that out of the box or the worm, I suppose. So what kind of strategies do you think you'll have to employ to try and include some of these more fragmented or difficult data sets into your system?
Yeah, it's a little bit open-ended. I mean, we know because we've done this work in terms of the nuclear positions about how much consistency, and it's pretty consistent. There is in the nuclear positions among multiple data sets. We've quantified that. So on that level, it's not a major issue. There's a certain amount of variance which has to do actually more with the variation in the temporal scale of the development, which is very sensitive to temperature. What no one really knows, including us, that we're about to find out, is on the level of the neural morphology, how consistent things are. We know they're quite consistent, and we also know that you're not going to see like the most fiddly of processes that will probably not quite line up from embryo to embryo. So we think at the level of detail we're going to be segmenting, that if this is pointing over here, this process is pointing over here, and this one is pointing over here, at the level of detail of the segmentation we're doing, they're not going to be like this if we take them from two different embryos. But it could be that they do, in which case some local warping is going to have to be developed to try and compensate for that.
Question. If you'd like to go to the microphone, because I guess it'll be on the video, so that'd be helpful. Thank you.
Same goes for when you give your answer.
Yeah. So your lineage seems to be running out round about 500 cells, and I assume that's because that's when the embryo starts moving.
It gives out a little bit earlier than the embryo starts moving. There's probably another 30 minutes after the screenshots I was showing. We're actually about to integrate another data set that brings it within like 10 minutes or so. The limiting factor is, to my mind at least, the microscopy, because you wind up sort of, People always say you can track everything in the worm community. They tend to say that, but when they mean everything, they mean 80 to 90%, and you spend a sort of exponential amount of time discovering which the 10 to 20% you can't track are. So we're pushing it to a later data set right now, actually. It's just hasn't been integrated with the app yet.
But no plans for after it starts moving?
We've created some preliminary plans. The imaging is fast enough to sort of freeze frame it even when it's moving, but that doesn't mean the image quality is quite good enough to see where everything is. You know, motion blur is not what you're so much worrying about. You're worrying more about just how small and how fiddly the morphology is and how easy it is to lose one cell up against another, even to your eye.
Okay.
Okay, any more questions? No? Okay, so just leave me to thank Anthony. Thank you very much.
Thanks.